When I was last week preparing what to say for this event, I went through the schedule and decided that I'd like to address a few a kind of general medical issues, medical and public health issues that pertain to the eventual use of nanotechnology. It's obvious from our talks we're at a very early stage of the field, and later today we're going to hear from the inventor of one of the first nanotechnology projects. So there's a great kind of future ahead. And it's a very interesting intersection between biology, medicine, life sciences, and engineering. And it's interesting to contrast the different approaches that engineers have to that which biologists have. Biologists are immersed in in many ways, very different kind of system. Engineers have a different approach based on believable physical laws, whereas in biology we have a little problem coming up with general laws that pertain all the time and everywhere. We tend to be a little more statistical, probabilistic. Now, the biological systems have, in a way, been defined as systems in which you have diversity and then selection and evolution. And life matter has a genome, the human genome, for example, which has a great deal of conservation in it. That is, many of the genes in our genome, 70, 80% perhaps, are strongly conserved. That means we contain within our own genome genes that are very similar to those that archaea had. That is one of the most primitive forms of life that shares about a third of its genes with humans. That's really quite remarkable. And it's these genes, the conserved and occasionally introduced new genes, that are the subject, that's the material on which evolution is based. That is, when changes in the genome required because of changes in the environment, they select these ancient genes. It's really interesting to question why we've retained all of those. And you can't help feeling that if in the explanation of how this might happen is that we can't predict what happens in the future. So we have genes that are prepared to deal with an unknown and unknowable future, but which reflect much of which happened in the past. Makes you think there's a kind of cyclic character to events. But in any case, the genome and consequently the phenotypes, the things that make processes work in biology, is not built for efficiency at any given time, because it retains these genes and therefore the products of the genes that may be useful in the future. So we're less than fully efficient. On the other hand, engineers want to design devices that are highly efficient. And they have everything that they need, but not much more for what they don't need. There is some evolutionary thinking in engineering, but the kind of time scale we're talking about precludes a great deal of concern about that. So biological systems are a lot less efficient than engineers would like them to be. There's always this remark that if I had to do it, I'd do it another way. And that may be quite true, but that doesn't take into account the great time depth where that happens in the evolution of species. Now, there's another feature of designing therapeutic diagnostic methods which kind of looks at the potentially the putatively affected population, that's the people who get the drugs, as homogeneous, whereas everyone knows they're not. And this issue has really come to the fore in the last decade or so because of our greater knowledge of the genome. And we recognize that there's enormous variation within the genome. Much of that is now recorded in the SNPs collection. Those are collections of polymorphisms. That is sections of the genome where variation can occur or does occur. And those SNPs, polymorphisms, are now widely used to determine differences between individuals that relate to susceptibility to disease and also different responses to drugs. So it's been, in my experience, very hard to get this well within the thinking of drug companies who don't like the idea that there may be certain parts of a disease group that are not susceptible to, that are not treatable by the drugs that it had. In effect, it kind of decreases the market. To my view, this is rather short-sighted because it means you can identify a portion of the population that is highly, will have a very good response to the medication, and then by presumably possibly some minor variations in the formulation or in the chemical structure of the drug in hand, you can make it better for the people who don't respond to the initial medication. So in all these therapies, we're thinking about the preventive measures, the treatments, devices. It's important to keep in mind that you can essentially say for any form of treatment or prevention, there'll be some people who respond and some who don't. When people are exposed to a infectious agent, let's say, some get sick and others don't. It's one of the most amazing, interesting aspects of medical science. And I spent the first, 20 years of my scientific research on that very issue. Back in those ancient days, and we're talking 50 years ago, we didn't have a genome to use to study variation, but we used the phenotype. That is, you could study certain proteins and look for variation. But instead of, currently by examining the databases for the genomes, you can find 2 or 3 polymorphisms before T. But it would take 3 years to establish you had found a polymorphism using involved family studies and so forth, when we only had the phenotype to deal with. So again, it's very important to keep this in mind when thinking about any of the potential therapies that will come forward in the future. Now, another possibility of nanotechnology is very increased sensitivity. That is, you can detect abnormalities, potential abnormalities, at a very early stage. That's both good and bad. The fact is, for many diseases, many cancers, people may have indications of cancer at a very early age. And the argument would be to cut it out surgically or provide treatment for it. While it's known that many of these cancers may remain in the individual, may grow somewhat, but they'll die of something else. That is, the rate of growth is such that the competing risks, i.e. other diseases, will kill the person before you have to worry about the cancer. And what you don't want to do, if it's possible, is to fuss around, remove the cancer or treat the person if he or she doesn't require it. Now, a case in point, which many of us are more than mildly interested in, are cancer of the breast and cancer of the prostate.